Hello, everyone. This is Cynthia, your classmate in biotechnology and society. It's my great pleasure today to present you with my research for the course on mitochondrial replacement therapy. Now, unfortunately, I'm a bit of a dinosaur with the computer technology here, so I haven't been able to figure out how to get rid of the left-hand column, but you can focus on the center and take a look at my slides as we go along. It's probably going to be helpful for me to have that left-hand column because I'm not really used to talking into a computer to teach. As some of you may know, I am a philosophy professor at Southern New Hampshire University, so this project is particularly helpful to me. Next summer, I'm going to be teaching again an ethics course in which is a component for bioethics. And I'm going to make this project a centerpiece of that bioethics. We needed a topic that really covers a lot of ground, and I think this one is important on so many different levels, as we will see. So let's go right to the beginning then. If I could figure out how to do this. Yes, here we go. Most of you, I'm sure, are aware that the traditional way of making a baby requires two people. You can do it in the natural way, or thanks to assisted reproductive technologies, you can do it in a petri dish. But either way, you have to have the DNA of two human beings joined to create a entirely new, genetically new human being. Well, that's the way it's been up until recently. Meet Dr. John Zhang of the New Hope Fertility Clinic in New York City. Dr. Zhang is holding a newborn baby boy who has the DNA of three parents. Why would we want to bioengineer a three-parent baby? And how would it even be possible? So let's take a look at these questions one at a time. I'm sure you remember from your basic biology that the cell has these organelles in it. Amongst these organelles, you have well, first of all, you have the nucleus in the center where our DNA, nuclear DNA, is contained. And then you have, amongst the organelles of the cell, the mitochondria. So you see in this picture here, you can see little red squiggly things. These are the mitochondria. The mitochondria are extremely important for us because through a process called aerobic respiration, they convert oxygen and nutrients into ATP. ATP is the chemical that gives us energy. It powers our battery, so to speak. But the interesting thing for our purposes here is that mitochondria have their own DNA, and they reproduce independently in the cell. We think this is a result of an evolutionary endosymbiosis that took place way, way, way back in the past. Some prokaryotes consumed some primitive mitochondria, but instead of completely digesting them, the two organisms developed the symbiotic relationship, and then they continued along together. So you have in our cell the nuclear DNA, but you also have these tiny mitochondria who are the powerhouses of the cell, who have their own DNA and who produce independently of the cell. So looking further into this picture, okay, here you can see in this picture, you have this time they're white, the mitochondria. And you look in the nucleus, you see the chromosomes, which I'm sure you know have the nuclear or the DNA information. That DNA information will be responsible for passing on your familial traits. If we look closer into the mitochondria, what we discover is that they have their own organelles. But if you see these purple squiggly lines in there, those are actually the mitochondrial DNA, and they're round, as opposed to the linear DNA of the nucleus. So I found this explanation of the differences here between these two kinds of DNA. You know, nuclear DNA you're going to get from both your parents. But what's interesting about mitochondrial DNA is you only get it from your mother. This is why anthropologists can use mitochondrial DNA to trace ancestry and the movements of human populations, because that mitochondrial DNA from the mother can be traced all the way back. Nuclear DNA is linear, as we said. Mitochondrial is circular. Look at the difference in amounts here. Your nuclear DNA, 3.2 billion base pairs. Mitochondrial DNA, just, around 16,000 base pairs. And whereas you have 20,000 genes in your nuclear DNA, you only have 37 genes in your mitochondrial DNA. The other important thing on this list that we want to look at is that nuclear DNA has a lower mutation rate, whereas mitochondrial DNA has a very high mutation rate. And it's for this reason that we have a problem called mitochondrial disease. When the mutations to the mitochondria are serious enough, they will impact all of the things for which mitochondria are responsible, the regulation of our metabolism. This disease will especially hit those parts of our systems which are highly metabolically charged. That would be the brain, the heart, the muscles, the nervous system, and they also seriously impact respiration. So if we go back to our friend Dr. Zhang here holding this little boy with the DNA of 3 parents, this little boy's mother had a mitochondrial disease called Leigh syndrome. She had already suffered 4 miscarriages and had lost 2 live children due to Leigh syndrome. And what's really tragic here, too, is that the mother is carrying this mutated DNA, but she is not impacted by it. She's healthy. It's her offspring who will be impacted. I could have chosen many, many examples, but I was really touched by the story of this little boy here, Grayson Baker. He's still alive. He's 19 months. He was diagnosed with Leigh syndrome. He's basically helpless. He has to have people do everything for him. He depends on machines to breathe and to eat. He was in the news recently because his mother wrote a heartfelt letter to Vice President Pence about the changes in the Affordable Care Act. He's one of the central figures in a foundation for mitochondrial research. You would be amazed. I was amazed at this statistic. 1 in 2,500 people are affected by mitochondrial disease. So this is not something which is a peripheral medical issue. So there's no cure for this disease. However, we now have the technology to eliminate the possibility of the disease right at the start of life. And this is called mitochondrial replacement therapy. There are 2 ways that you can do this. The first way is called pronuclear transfer. Basically what happens here is you take an egg from the mother and an egg from the donor woman. That would be your eggs here lined up on the left side of the screen. The tan one is the mother, the blue one is the donor. And in this, you fertilize the eggs first with the sperm from the father. And then you remove the nucleus, the nuclei, from both fertilized eggs. You cast aside the nuclei of the donor woman, and you insert in that place the nuclei, the pronuclei of the mother. What this means is that the cytoplasm, where the mitochondria dwell, is completely from the donor woman. It is healthy. It is not mutated mitochondria, yet you still have the DNA of the mother. So this zygote now has the DNA of 3 people: the father, the mother, and the mitochondrial DNA of the donor woman. In spindle transfer, basically what you do is, instead of taking the whole nucleus, you take, as you know, the chromosomes have spindles attached to them. So you basically just grasp the spindles and the chromosomes and replace them into the donor. The biggest difference here, of course, is that these are unfertilized eggs to start. Once the spindles with their chromosomes have been placed in the donor woman's egg, then you have the fertilization with the father's sperm. Now, this mitochondrial replacement therapy is here to stay. This is a picture of another group who are working simultaneously in the Ukraine. They are holding a baby girl who was born on January 5th of this year. While this child was, at the time of this child's birth, another woman in that clinic was due to deliver as well. Dr. Zhang, who we saw at the start of this presentation, his clinic has planned 20 babies for 2017. I wasn't able to follow up to see if those 20 babies came to be, but they were definitely in the business of doing MRT henceforth. It's interesting to note that the procedure is illegal in the United States because, if you think about it, you're affecting the germline. The germline is the material, the genetic material that is passed on in a family. And in the United States, we have laws prohibiting experimentation or alteration of the germline. So in order to do this, even though Dr. Zhang's clinic was in New York, he had to do this procedure in Mexico. This technique, this bioengineering technique, has significant repercussions. For one thing, it can be a dream come true for lesbian couples. Usually if a lesbian couple wants children, one of the partners has to carry the child that she conceives through in vitro or through, I can't remember it right now, but their child will not be genetically linked. This will allow two women to have a baby. They'll have to find a single father to fertilize, but the two women will have a baby sharing their genetic identity. That issue alone shows you how controversial this thing can be. First of all, just taking a look at what we're doing to the DNA itself, okay? You're bringing together the nuclear DNA of one woman with the mitochondrial DNA of another. So you're permanently changing a germline. This is going to be very serious for people who do genealogical research or anthropological research. They've always depended on the maternal mitochondrial to trace ancestry, because we only get our mitochondrial DNA from our mothers. It's something passed down from mother to child, mother to child. So we trace genealogy and the movement of races and populations through this maternal mitochondrial DNA. We're now tampering with that, so we won't be able to do that anymore. The procedure is so new. We really don't know what the long-term effects are going to be on offspring or the offspring of offspring. We're concerned about something called heteroplasmic drift. This is because, as we said earlier, mitochondrial DNA mutates very, very easily. So if we're messing around with it, okay, we could create new mutations, if not now, then down the road, and that would basically undo everything that they're trying to achieve with MRT. Psychologists have tons of questions about this. How is this going to impact our sense of ourselves, our self-understanding? What is it going to mean to belong to a family? You can even see the legal issues that are going to come from this, that could come from this, if someone claims to be the third parent and has rights. We have no laws on the books to deal with this at all. And finally, it's special interest groups, like we saw with our lesbian couples. They can seize upon this for non-therapeutic purposes. But I think the more dangerous notion is that eugenicists could get involved, because, as you know, eugenics is that belief that we can purify the human race, and they could make use of this technology. Another thing that I didn't get to put on the slides, but I think I should mention, is that it can also be used by older women wanting to have children. As we get older, our mitochondria get older too. And if a woman wants to conceive a child in her late 40s, she could probably look into this therapy to make sure that her child is getting her DNA, but the cytoplasm would come from a donor, guaranteeing that the mitochondria for the child would be strong and healthy. So that pretty much brings me to the end of my presentation. I think this is an extremely important topic for bioengineering, for bioethics, because it touches so many different areas of human life. It's here to stay. It's not gonna disappear. It's gonna become part of a regular assisted reproductive technology. So thank you very much for your attention. Bye-bye.